Hello all, my name is Ryan Abdelaziz, answering the initial post for VT3. The positions are: what is the most underappreciated challenge in managing big data for biotechnology research? Why is it persisted despite rapid advances in computational capability? Should experimental design drive how data pipelines are constructed, or should available analytical infrastructure determine what experiments are even attempted? And is more data generally an asset or a liability in hypothesis-driven biotechnology research? Where does data abundance help, and where does it risk obscuring meaningful data? So I think that the most underappreciated challenge in biotechnology big data is not storage or computing power. Rather, it is preserving the biological and clinical meaning of the data from collecting through analysis. So this is especially difficult in psychiatric research, where phenotype is often reconstructed rather than directly measured. Clinical history may span years, multiple institutions, changing treatment protocols, different diagnostic eras. Certain records created using previous diagnostic manuals, DSM-IV, may not fully be equivalent to those interpreted under DSM-V. In postmortem research specifically, investigators may also rely on medical records, toxicology, medication histories, and next of kin phone screenings. When these screenings questions or adjudication procedures change, the same symptom may not be measured consistently across donors. That is sort of my position for the first prompt. Most underappreciated challenge is loss of interpretive context. It persists because algorithms cannot fully recover information that was never collected, was documented inconsistently, or cannot be harmonized across institutions and time. So a dataset may contain millions of variants and gene expression measurements, yet the main clinical variable may remain uncertain. In studies of MDD or an apparent major depressive disorder, an apparent molecular difference could also reflect ancestry, medication exposure, brain region, postmortem interval, RNA quality, cell composition, or sequencing batch. Statistical model may be technically correct, while the biological interpretation is incorrect. So this, for the second prompt, my position is a design first but infrastructure aware. Experimental design should determine how the data pipeline is built. Research questions should define the experimental unit, sampling strategy, metadata requirements, quality control criteria, blocking structure, and statistical model. In psychiatric brain research specifically, that means deciding in advance how diagnoses will be defined, how older records will be reconciled with current criteria, which covariates are essential, and how uncertainty in clinical narrative will be representative. So infrastructure still matters. A study is not rigorous if team cannot securely store sensitive data, document transformations, or reproduce the analysis. However, infrastructure should constrain feasibility rather than dictate the scientific question. Existence of a sequencing pipeline should not be the reason an experiment is attempted. For the third prompt, I do not think that more data are automatically an asset. The important distinction is between more measurements and more independent information. More data help when they add biological replication, represent meaningful heterogeneity, or connect multiple levels of a coherent mechanism. More independent donors can improve precision. Better clinical records can strengthen phenotype definition. Matched genetic, transcriptomic, epigenetic, and clinical data can help connect genetic risk to disease biology. So more data becomes a liability when volume substitutes for design. Millions of molecular measurements do not compensate for small or poorly characterized donor cohort. Deeper sequencing does not correct inconsistent diagnosis. Repeated measurements from one donor do not become independent biological replicates. More variables also create more processing choices, quality control thresholds, and statistical tests, increasing the risk of unstable or confounded findings. This is essentially important, again, in biotechnology, and specifically in psychiatric research, because molecular assay may be more precise in the phenotype. Sequencing can be highly standardized, especially when clinical narratives were assembled across different sequencing can be highly standardized, yet clinical narratives can be assembled across different institutions or systems, screening protocols. So if that uncertainty is ignored, molecular precision can create false sense of confidence. So kind of to connect the 3 prompts, the central challenge is preserving interpretive context. Experimental design should drive the pipeline, while infrastructure should support feasibility. More data are valuable only when they add independent, well-characterized information. Big data should therefore be judged by information quality rather than volume. Computation can amplify a rigorous design, but it cannot restore clinical meaning once that meaning has been changed or lost. So yeah, that sort of answers my initial thoughts on the VoiceThread 3. I hope that you guys find my discussion interesting. I'm looking forward to hearing any sort of remarks or comments you have, and looking forward to discussion later on. Thank you all.